So Vincent, is this tool recorded?
Yeah, so we're going to record it, and it's also being live streamed on YouTube. And then we'll put the video on our website. And so welcome to the 784th monthly meeting of EPAASM. Today we have Dr. Isabel Copeland from Johns Hopkins. So first of all, just to remind you, for the Zoom, if you want, we'll hold all the questions toward the end. And if you have questions, please tap the participant tab, and you can press the blue hand button to ask the question. I will unmute you so you can ask the question directly. Alternatively, you can type the questions in the chat, and I will relay those questions. Dr. Copeland is the Asm Distinguished Lecturer. This program has been going on for more than 55 years and more than 600 plus lectures. It's provided by the National ASM to the branches, and it's being funded by the Whitman Foundation for Microbiology. And so please join ASM, which provides you the community as well as the network and a lot of career opportunity and a lot of science that's available both online as well as general meetings and journals. And of course, when you join ASM, also join the EPAASM chapter. We've been going strong for over 100 years now. So today it's my pleasure to introduce Dr. Isabel Copeland. She received her PhD from the University of Louvain in Belgium. She received her master in drug design from the University of Lille. And she postdoc at the Institute for Cellular Pathology, the Pasteur Institute of Lille, as well as the Yale University. She and her lab has been working on and made a lot of seminal discovery on the cell biology of host-parasite interactions, particularly how the parasite can modulate the vesicular trafficking as well as savaging host lipids to support replications and differentiations. She's co-authored over 120 articles, numerous reviews, as well as book chapters. She received a lot of prestigious prizes, including the Pierre Edouard van Beneden from the Belgian Royal Academy of Sciences, recognizing her outstanding work in cell biology. The prize from Rome World Foundation, recognizing her work on the cholesterol metabolism. She's also received both the Faculty Innovation Award as well as the Discovery Award from Johns Hopkins. So without further ado, thank you very much for accepting our invitation. She's going to talk about catching what you eat, the secret of intracellular tenure of Toxoplasma gondii.
Thank you very much, Vincent, and also the ESM. It's a big honor. I'm a member of this society since 2006 and still very charmed by all the topics that this society is organizing. And I'm particularly motivated today to present my talk because it will be on parasites, the parasite Toxoplasma, and I really want to boost somehow the topic of parasitology, both protozoan and elements for this society, mainly focused on bacteria and virologists, just to expand this horizon. Okay, so I like to start my talk by showing these very nice pictures. So in Greek mythology, Cerberus is this monstrous three-headed dog that guards the entrance of the underworld, allowing the dead to enter, but devouring those who want to escape. And I had this parasite is in green in this pseudo-coralized electron micrograph.
Sorry, Dr. Copeland, you have to share your screen again to show this slide.
Okay, so I will do again. Is it visible?
Yep.
Okay, wonderful. So I was just saying that Cerberus that we can see in Greece, that I had, of course, represented as a toxoplasma, is a monster. And this parasite is, as you can see, live in the vacuole, called the Parasites vacuole, in the cytoplasm of different mammalian cells that you can see in green. And I have symbolized toxoplasma as a Cerberus, this monster, because you will see that this parasite, despite being secluded within a vacuole in the cytoplasm of mammalian cells, is very proficient in scavenging and consuming host cells material, as you will see. Okay, so let's start by the disease. We contract the parasite Paros by ingesting the cyst form present in the food, the water, or non-cooked meat. Then the parasite from the GI tract will invade epithelial cells of different gut tissue, where they will multiply for a couple of days. That is what we call the acute infection. From them, the parasite will reach the lymphatics and the blood vessels to reach different tissues, including, preferentially, the brain, the eyes, and the muscles, including the heart. And in this tissue, they will form cysts, kind of semi-dormant form, and that is what we call the chronic infection. If you are immunocompetent, you will not realize that you have cysts in these tissues. The pathology starts when you had a reactivation of the cyst in situ that can lead to necrotic tissue when you have the parasite. In case of heart infection, you can have cardiac toxoplasmosis, so arrhythmia. Eyes, you can have blindness, but the more dramatic situation is when you have a reactivation of the cyst in the brain that can lead to encephalitis. Another pathogenic situation is when the mother is contracting the parasite for the first time during pregnancy. The parasite can cross the placenta and reach the brain of the fetus, and that can lead to different congenital situations, including brain defect. So we can say that toxoplasma is certainly one of the most world successful parasites for many reasons. First, it can infect nearly 1/3 of the world population. So because it's so easy to be contaminated by ingesting the cyst form of the parasite. Then the parasite can live inside all warm-blooded animals, so the reservoir is huge. In our body, is equipped to control and circumvent or attack from the immune system. For example, the lysis of the host complement cascade by secreting different serine protease inhibitors. It can invade any nucleated cells, so very cosmopolitan, no cell specificity. It can avoid also toxic oxygen metabolite during entry. And when it's penetrating the cells, it's forming a unique vacuole, the parasitophus vacuole, that is a self-made niche that doesn't contain protein from the host cells and that can also be recognized and destroyed by host lysosome. Then in our body, the parasite can differentiate into semi-dormant form in the brain and in the muscles, protected by a thick cyst wall derived from the vacuolar membrane that is impervious to immune attack. Well, despite this obvious success feature, the parasite has some weaknesses. For example, he lost many metabolic pathways. He cannot synthesize a lot of important nutrients like lipids or amino acids. He became what we called an obligate intracellular parasite, meaning that he cannot survive extracellularly. It implies also, to survive, that he needs to scavenge many essential metabolites from the host, implying that he must synthesize all the proteins, transporter, and enzymes needed to salvage and store nutrients. And this is all the time. But besides its weaknesses, our lab wants to identify more. And in this cartoon, I have summarized all the metabolic deficiencies of the parasite. In pink, I have highlighted all the molecules that the parasite cannot synthesize and need to scavenge. In blue, those that can be scavenged or made by the parasite. And I think it's very important because you think about generating a drug that can block the pyrimidine biosynthetic pathway. The parasite will grow fine because it has transporter to scavenge host pyrimidine. So from this cartoon, how can we think about affecting the parasite replication? A first way will be, of course, to cut the source of this important metabolite. We have identified that cholesterol, the main source of this lipid for the parasite, is plasma low-density lipoproteins. If you cut the source of lipoproteins, for example, in a medium depleted of these lipoproteins, or if you block the egress of cholesterol from lysosomes, the parasite will stop growing. Another way to affect the parasite growth will be to substitute this important metabolite with analogs that are, of course, not toxic for mammalian cells. For example, if you replace in the medium cholesterol by phytosterol, the parasite will not make the difference, will incorporate this sterol in their membranes, and that will induce, of course, membrane damages. We can also replace choline by its analog, dimethyl-ethanolamine. The parasite will scavenge dimethyl-ethanolamine to synthesize phosphatidyl-dimethyl-ethanolamine instead of phosphatidylcholine, and again, membrane damages. I will tell you that we have an enzymes that can convert phosphatidyl-dimethylamine into a phosphatidylcholine. So these metabolites are absolutely no toxic for mammalian cells. Another observation that we did is that the parasite has a greedy appetite for some nutrients. For example, for unsaturated fatty acid oleate. More you introduce these lipids in the medium, more the parasite will take. As a consequence, it will be completely engorged with fatty acid and it will be putting at risk of lipotoxicity. I will show you that on this cartoon very clearly. After 1 day, the parasite forms this beautiful rosette in the cytoplasm of mammalian cells, controlled condition. If you add oleic acid at physiological concentration compared to the concentration in human serum, you can see that the parasite don't divide. It's completely engorged with lipids, puffed with this fatty acid. As you can see, they have this large lipid droplet, and definitively it cannot cross. So meaning that in our list of vulnerability of the parasite, we can add that it has a poor discrimination for host metabolite, and it has a greedy appetite that can be its downfall. Basically, for lipids, it acts like an adipocyte, but of course, it's a small size, and it's completely, let's say, full of lipids in case of oleate. So in the lab, we are studying how toxoplasma acquire nutrients from the host cells. Before entering into this topic, I want to summarize the main feature of toxoplasma infection at the cellular level. First, the parasite invades cells through a process called active invasion. So it can generate its own vacuole through lipids and protein that is secreted inside the host cells. And this process has nothing to do with phagocytosis. That is very important. Meaning that after internalization, it will be within this vacuole, like an incognito pathogens, and this membrane is not physiogenic. So no recognition with host lysosomes, no destruction through hydrolysis of the cells. So perfect protection directly after entry. Then the parasite will divide rapidly, and when the host cell will be completely filled with the parasite, they will need to ingress and invade new cells. So concerning the mechanism of nutrient acquisition by the parasite, at least we know one main phenomenon. We know that the parasite can modify the permeability of the vacuolar membrane by forming some pores made of proteins that allow the penetration of small solute from the host cytosol, like glucose, ATP, that can enter the vacuole. And also, our laboratory has identified that the parasite can also scavenge nutrients from host organelles. And that is a unique process, as I will describe. So why this scavenging? Well, these organelles can be a very important source of nutrient and very abundant. Mitochondria here can be important for lipids like phospholipids or cholesterol. Golgi for sphingolipids, endolysosome, for example, for many nutrients coming from the medium. And I will give you 3 examples why and how the parasites scavenge the Golgi, lipid droplets, and endolysosomes. Okay, coming for the Golgi, we were curious to know about the morphology of the Golgi upon infection. We have the parasite here in red, and in green we have the representation of the Golgi upon infection. Well, you can see that obviously the Golgi is fragmented, and we can count the number of Golgi foci in infection versus non-infected. But then looking at the electron microscope, in case of non-infected cells, the Golgi is forming these long crescenting organelles. When we have a vacuole containing the parasite, we cannot recognize this long shape of the Golgi, but instead, in yellow, you can see all these small mini stack of the Golgi cluster around the vacuole. And we have identified that is, of course, the Golgi through anti-Golgi staining. So why the parasite is fragmented the Golgi? Well, maybe it may need lipids from these organelles. We know that Golgi process ceramide to synthesize major sphingolipids like sphingomyelin. And simply, if you label the cells with fluorescent ceramide, the Golgi is labeled. Then when you have a toxoplasma that you can see here, you can always look at these beautiful flowers like the rosette of the parasite. You can see that the Golgi is again destructurated, organized all around the vacuole, and progressively with the pulse time, we can see that the parasite is becoming fluorescent. So obviously, it has somehow access to the lipids coming from the host Golgi. Another example is for lipid droplets. We could see that the lipid droplet, here stained in green, cluster around the vacuole. We can amplify this process by incubating host cells with oleic acid that stimulate the biogenesis of lipid droplets. And we can see a more massive clustering of lipid droplets around the parasite that we can also confirm by EM. All these white structures are lipid droplets. So again, we need to see why and how the parasites what is the purpose of this recruitment of lipid droplets. And for that, we had this protocol when we had cells that we have incubated with body P, this type of dye, that is actually a non-metabolized fatty acid. This dye will accumulate inside lipid droplet, but not be after that incorporate for the formation of other lipids. When we infected cells, in these treated cells, and observe the parasite by fluorescent microscopy, we can see a green signal. And this green signal is definitively these lipids coming from lipid droplets. We went further to see if lipid droplets are important for the parasite infectivity. And yes, in vivo, we have obtained from Rob Frazee in Harvard a knockout cells that lack DGAT1 and DGAT2, these two main enzymes in mammalian cells that cannot, in this case, there are no production of triacylglycerol. As a consequence, there is very tiny lipid droplet in these knockout cells. And our test to monitor the parasite growth is to count the number of parasites per vacuole. And we could see that in the knockout, there are very small vacuoles that are produced. That is the situation in vitro, but then in mouse, we can infect it with the parasite, knockout mouse lacking DGAT1 enzymes. Of course, the double knockout is not viable, and we can observe that the parasite form less cysts and they have small size. So lipid droplets from the host are important for the parasite infectivity in animals in vivo. A third example is giving for the importance of endolysosomes. And we have identified many studies ago that the parasite retrieve cholesterol from plasma lipoproteins. And to visualize this process, we have incorporated fluorescent cholesterol into lipoproteins. And then we can see progressively that the parasite is labeled. So retrieving cholesterol from these organelles. And we could see a beautiful gathering of LDL site endolysosomes in this electron micrograph. The LDL particle can be tracked with these gold particles. So the question that we have in mind, obviously the parasite is scavenging the nutrient content of these organelles. Because there are no fusion, what could be the process to acquire this nutrient? The first observation that we did in many laboratories is that the toxoplasma vacuole acts like a magnet. It attracts many host organelles, the ER that we can see here in green around the parasite in red, lipid droplets, the Golgi in green that is fragmented, endolysosomes, and also mitochondria. So it's possible that attracting these organelles, and we know for some of them, like mitochondria that is mediated by parasite proteins, will maybe facilitate the access of the content of these organelles. So to start to address this important issue, we want to know first which host trafficking pathway the parasite are targeting. So obviously, it needs to scavenge somehow cholesterol through the endocytic pathway. You need, of course, to scavenge sphingolipids from the biosynthetic pathway, the ER and the Golgi. So to try to define the point of intersection between the vacuole and different organelles, we track the distribution of RAP vesicles in toxoplasma infected cells. A RAP being, of course, like an organelle markers, with, for example, RAP11, shuttling the content of early endosomes to recycling endosomes. So to see which kind of pathway are selectively targeted by toxoplasma, we transfected mammalian cells with different GFP RAP construct and look at the distribution of RAP vesicles in toxoplasma infected cells. Let me give you first an example of RAP1, shuttling the content from the ER to the Golgi. So that is the pattern of RAP1 vesicles in non-infected cells. When we infect with toxoplasma in red and the vacuolar membrane is in blue, you can see that there is a complete delocalization of the RAP vesicles at the PV. And when you increase the time of infection, you can see that this gathering of RAP vesicles is increased. Another example from RAP11, coming from early endosomes to recycling endosomes and the plasma membranes, we have observed on this confocal view of the same vacuole, so different Z slices. If you look at the middle of the vacuole, in the middle of the rosette formed by the parasite, there is an accumulation of green foci. So that could be a RAP vesicles. So somehow the parasite managed to scavenge a host organelle intact that has to pass through the vacuolar membrane. Of course, the first control that we need to do is to be sure that this GFP foci corresponds to RAP vesicles and not an aggregate of GFP proteins. And for that, we have transfected cells with a GTP lock mutant of RAP. We know that RAP protein shuttle from a free form in the cytoplasm and a vesicle-bound forms, GTP locked. And in this case, by counting the number of vacuoles containing GFP foci, we have the GTP, the vesicular bound form. There is exactly the same number of vacuoles. Compared to the GTP lock, so free, you can see that there are no signal or very faint inside the vacuole. So meaning that these foci correspond to bona fide vesicles. So we transfect different GFP RAP constructs from different trafficking pathway, endocytic, cycling, anterograde, secretory, to conclude that by counting the number of PV containing these RAP vesicles, that the parasite intercept a lot of pathways, but with some selectivity. He intercepts mainly the recycling pathway, maybe to retain inside the cells nutrient coming from the environment, blocking the egress through the plasma membrane, and also the secretory pathway at the exit of the ER and of the TNG, so to acquire a lot of metabolites synthesized by the cell. Of course, we want to prove that this interceptions is linked to nutrient uptake, and for that, we follow the fate of RAP14. RAP14s go from the Golgi to the plasma membrane to deliver sphingomyelin. We have an infected cells. We can identify in the vacuole these RAP11 vesicles. Then we incubate cells with fluorescent ceramide, the parasite in label. And then by tracking only the signal, GFP positive and ceramide positive in yellow, we could see a gathering of these RAP vesicles around the PV, but also inside the vacuole. So these RAP vesicles are loaded of lipids, nutrient for the parasite. Okay, so somehow the parasite managed to attract and scavenge and sequester host organelle inside the vacuole. And the question now, what kind of pathway or mechanism? And that leads us to introduce you a very interesting structure, an intravascular network that is mediating this process. So if you do electron micrographs of freshly egressed toxoplasma, you can identify in the cytoplasm a strange organelle, a kind of vesicles containing tubules at different stages of formation and thickness. Then looking at this parasite, we can see that the parasites secrete this membranous structure inside the vacuole. We have here different parasites, and we can catch here a process of secretion in block of this membranous tubule. Then indeed, if you look at low magnification, this vacuole containing 8 parasites, we could see all these tubules within the vacuole between the parasite everywhere. And we call this membranous tubule the intravascular network, or IVN. I will show you another example. Here we are in the middle of the parasite, of the vacuole. And if you zoom, we can see beautifully these tubules. It's called the spaghetti between us. And why I attract your attention to this network is that we had identified that host RAP vesicles accumulate in the area when you have this network. This network, we don't know about the function, but at least we know that they contain proteins that can be very good markers, like here in blue or in red. And we can see that in green you have the accumulation of these RAP vesicles when you have this IVN. Okay, so what is happening? When you have an enigma of this amplitude, I like to look at this beautiful work from this Brazilian group, the Souza. They are performing beautiful scanning EM of different parasites, including toxoplasma. You have here a beautiful rosette of the parasites within a vacuole. Here you have the host cells. And through this technique, they can resolve the ultrastructure of the membranes of the IVN. And clearly, you can see that some of these tubules are connecting or are connected to the vacuolar membrane. And we are lucky by transmission EM to see these tubules appended to the vacuolar membrane. You have here the vacuole, the host cell, the parasite. And you can see that this appended tubule coming from the IVN can be very long. So what does it mean? It means that the parasite, by exporting this tubule to the vacuolar membrane, of course there's a process of fusion, has created some kind of open gates with the host cell cytoplasm. When we incubated the cells with LDL gold particles to track endolysosomes, we could see in the lumen of these tubules gold particles within a vacuole. So somehow host organelles are trapped inside these tubules and they are sequestered inside the vacuole. So it's a very unique process, of course. Of course, we want to prove that this network is involved in host organelle scavenging. For that, we make use of a knockout that, like this IVN, but instead form this unstructured walls of membranes, because these protein grade 2 are involved in the tubulization of this network. And indeed, by exploring this knockout, also a double knockout, so this protein is also involved in the stabilization of the network. By counting the number of GFP foci compared to the wild type, you can see that the knockout are almost empty. They have a reduction by 80% of the number of RAP vesicles in the lumen of the vacuole. Okay, so what I show you is, of course, static picture by IFA or by electron microscopy. But what is important is to deliver a dynamic view of this process. And for that, we have engineered a parasite cell line expressing a protein and cherry in red that label the vacuolar membrane, but also the network. And we had infected cells expressing a GFP RAP11 vesicles. We can see that the RAP vesicles in green are in the cytoplasm. They are moving extremely fast. And then you can see in the movie RAP vesicles in yellow, because inside the tubules that move slower. You can see here on this movie. So this is the host RAP vesicles trapped inside the vacuole when you have the location of the IVN. So that is really the gate of entry inside the vacuole. All right, so another step that we have observed into this process is that if you believe that the tubule formed by the IVN attached to the vacuolar membrane are the door for this organelle to enter, we observe that at the end there is an accumulation of host RAP vesicles in the middle of the vacuole. And that implies that there must be a process of scission of this tubule or detachment from the vacuolar membrane. And when you have this kind of formation of a vesicles through an invagination, we have either an involvement of the dynamine-like machinery or the escort machinery. The dynamine, of course, like we have a coated pit that become a coated vesicles, or for the involvement of escort machinery, formation of intraluminal vesicles inside a multivesicular body, for example. The parasite has, of course, dynamine and escort protein, like any eukaryotic organism, but it doesn't secrete this protein at the vacuolar level. So meaning that if there is a process of scission, we believe that the parasite may usurp the machinery escort of dynamine of the host cells to induce the scission of this tubule and then the trapping inside the vacuole. So that is the organization of the escort system, very, very sophisticated. And the main component of the escort tree that form long filaments inside vacuole to generate at the end a constriction prior to the scission is CHAMP4. And CHAMP4 shuttled from a closed and inactive form to promote this process of scission. And the first assay that we did will be just to transfect the cells with CHIM4B, GFP of MRO TAC, and look at the distribution of this protein in toxoplasma infected cells. And the result was spectacular. We have in blue, here there's two vacuoles. When you have a TAC at the C terminus, we have two vacuoles. In blue is the staining for the vacuolar membrane, and in green is the signal road for CHIM4B. The recruitment is massive, and as you can see here on this yellow signal. If you have the TAC at the N terminus, we have another pattern. We have an accumulation of CHIM4B when you have the IVN of the parasite. And this will be beautiful system to identify for this first tag, parasite protein located at the cell surface that recruit this escort machinery to induce the scission of tubules inside the vacuole, and also the second construct to identify parasite proteins that will mediate this scission. So we have beautiful tool because at the end is to find unique parasite proteins as a drug targets. We were curious to look at electron microscopy, the morphology of the vacuole under this condition of overexpression of Chim4B and TAG. And then we could see that there is a massive deformation of the vacuolar membrane. That is a magnification. We can see that there is this huge tubule formed with a striation. We see the signature of Chim4B, meaning that that is the step of construction that we are looking for. And then that will be remarkable to identify the composition. There is the parasite protein interacting and recruiting Chim4B that we have confirmed between Chim4B by immunoelectron microscopy. And also, if you want to know, if you incubate liposomes with a human version of I don't know which component of escort 3, we have also a formation of constriction and striation of this liposome, exactly what we have here with toxoplasma. Okay, so this is the step of internalization, and we are investigating the machinery. So what could be after that? The processing of nutrient inside the PV, because they're still inside organelles. And we have identified a potent lipase, actually a phospholipase 2, called LCAT, that are secreted in the parasite inside the vacuole. And this enzyme is cleaving a phospholipids by removing a fatty acid chain. As a consequence, we have lysophospholipids. And through its canonical shape, lysophospholipids act like a detergent, creating a destabilizing membrane, I would say. And we had identified that these enzymes, LCAT, are actually localized to the IVN. And we have RAP vesicles. We could see that the signal of RAP, the presence of RAP vesicles are all surrounded with this membrane, but you have this lipase. We have generated a knockout parasite lacking the lipase or overexpressing the lipase, and we could see that there is a reduction of the number of RAP vesicles inside the vacuole that we presume likely like a result of consumptions. So we can see that, wow, if the parasite is secreting a lipase, what could be the damage for itself? Well, we think that there is a kind of a level of protection because unlike other LCAT, the parasites secrete these enzymes into 2 fragments that share a part of the active site that need to be reassembled in 3D. And the parasite, by secreting these 2 fragments that need to be dimerized to reconstitute the active sites, can protect itself. So it can control when these enzymes will be assembled and so exposed to the, in this case, host organelles, to destroy the membrane of host organelles. Because that is indeed our working hypothesis. The parasite will invade mammalian cells. They have this pre-IVN. When they are inside the vacuole, they will secrete these tubules that will fuse with the vacuolar membrane. Up directly, small host organelle will be trapped into this invagination. Then we believe, to avoid, of course, that these organelles are coming back to the host cytoplasm, it will recruit the host escort machinery that will induce the scission and the accumulation of host organelle inside the PV. After that, the parasite can secrete this lipid that will make hole in the membrane of host organelles, and that will allow nutrient to escape and to be transported to the vacuolar, to the parasite, through the mini transporter at the level of the plasma membrane. So that is our hypothesis. Of course, you may have the question is that, oh, that is also an important slide. We want to identify the parasite protein involved in the binding and recruitment of host organelles and also the processing. We have generated this stable virocell line expressing RAP11 and looking to different parameters for this internalization and morphology of GFP foci using velocity software. The number of foci per vacuole, we can see it's about 200. The volume, the shape, the distribution inside the PV. We want to compare different chemical-induced mutant of toxoplasma with the wild type to identify this many parasite effector involved in this process. Okay, the questions that we can have also, how can the parasite lacking an IVM, I can say, and growing perfectly fine in vitro, can get its nutrients? And that brings us to investigate a more property of the vacuolar membrane. So that is this knockout. They had this kind of walls of membrane in these organelles. And if you look at the vacuolar space, it's almost empty. But you can see that the parasite divides. Okay, we make more staining of the vacuolar membrane. And it has been known by us in many groups that this vacuolar membrane form a long extension into the host cytoplasm. And for the wild type, about 2. And then this extension will increase until the time of the parasite division, 8 hours post-infection, and then they will decrease and disappear. But when you have the knockout, wow, we can see that there is a lot of extension in host cytoplasm. And this extension continues to increase until the parasite egress. And if you stain a host cell, for example, for an organelles, because we want to know there's some kind of interaction between this projection and host organelles, here in the case of lipid droplet, we could see that these vacuolar membrane projections completely encircle lipid droplet like a lasso. And by EM, you can see actually there's some kind of ribbon strangulating host lipid droplets. And we think, wow, maybe there are this extension. And to prove that, we did immuno-EM with a marker for the vacuolar membrane. And indeed, we could see that these ribbons are made of parasite materials. And these pictures are very reminiscent to membrane contact sites that we have in mammalian cells. So what are they? These membrane contact sites are this zone of close apposition between 2 organelles, one donor, let's say, and one acceptor. And so very close apposition, about 20 nanometers, made of structural proteins, tethers, that link the 2 organelles together. And also there are, of course, different transfer proteins for lipids of ion that shuttle the content from one organelle to another. And looking by EM, here's the wild-type parasite. You could see that the parasite that recruit host ER could see this ER element closely opposed to the vacuolar membrane. And that is the classical textbook. We look at the small space of a membrane contact site. So you are curious to know if the parasite can hijack membrane contact sites from the host cells to actually get the lipids from these lipid transfer proteins between 2 organelles. First, a tool that we did, we look at the distribution of VAP. VAP are important tether for the ER. And the ER, we know that they're contacting many organelles, mitochondria, lipid droplets. And these VAP proteins need to interact with protein on the donor of the organelles with a fat motif. And when we transfect cells infected with VAP, we could see a clear staining of the vacuolar membrane for these VAP proteins. And if you look at the knockouts having this long extension, here stained in red, we could see that the VAP staining is completely associated. So what we believe that a skill of survival for this knockout is that they form these huge extensions that contact organelles, and we believe that is to recruit, to catch the content of these lipid, let's say, transfer proteins from different organelles. So the acquisition of lipid here will be non-vesicular compared to the IVN for the wild type. We believe that when the wild type has this extension is also to recruit lipids via this membrane contact site formed with host organelles. All right, so the parasite needs lipids, of course, scavenge through vesicular and non-vesicular routes. Then we want to know what could be the next step. And it could be very important for a parasite oxotrophic for lipids to be able to store these lipids in case of shortages. So we know that toxoplasma, like mammalian cells, has lipid droplet, as you can see here. And lipid droplets contain cholesterol ester, generated through the activity of ACAT enzymes and triacylglycerol via DGAT. And the parasite has indeed one ACAT and one DGAT to store the lipid that it needs. We generate the knockout of ACAT, and we can see that the parasite is poorly in shape. They have this small body, and we have identified that the parasite scavenge cholesterol, although lacking these enzymes to store cholesterol, and they have a massive accumulation of free cholesterol at the plasma membrane, which is extremely toxic. We can see they are terribly affected inside the vacuole, and they have a poor growth. We test different, because the knockout seems very impaired in growth, we test different ACAT inhibitors, and we can see that the parasite is sensitive to some of them. So the conclusion, the ACAT enzymes can be a very good drug target for many reasons. First, it needs cholesterol and store, of course, in case of shortage, as I said, but also because it has no control, although no capability of storage, is still accumulating cholesterol without any restriction. Then we look at the other enzymes, DCAT, and the phenotype was even more spectacular. The knockout, compared to the wild type, has a very poor growth. As you can see here in green, the parasite after 1 day. After 2 days, the knockout, the wild type forms these large vacuoles, and the parasite start to egress, while for the knockout, is still inside the cells, it starts vaguely to grow. Then if you do plaque assays, after 10 days, we have this massive lyse of cells for the wild type and nothing for the knockout. And if you look by EM, they are still indeed inside the cells. So they grow very, very poorly. Then we try, because it's very spectacular also, a DGAT inhibitor, this one T863 that blocks DGAT1. And we could see that at concentration that I can tell you are absolutely not toxic for mammalian cells, that the parasite is extremely sensitive. If you look at the number of parasites per PV. And looking at the morphology also, you could see that even at 5 micromolar, which is nothing for mammalian cells, we can barely recognize the shape. So it's very, very severely damaged. So the next step was to see the parasites, of course, DGAT and ACAT enzymes used fatty acid as a substrate for cholesterol or diacylglycerol. And one student in my lab has observed that the parasite is a very gluten. It likes fatty acids, more especially unsaturated. And more she introduced fatty acid in the medium, where she can see that there is a massive accumulation of these lipids stored in lipid droplet in the parasite. And at this concentration of 0.5 millimolar, you can see that the parasite, you can recognize the organelles. It just has formed this huge, massive accumulation of lipids. So the idea will be, of course, to attack the parasite to 2 means, the DGAT inhibitors and the concentration of oleic acid that is deleterious for the parasite. You could see that individual treatment is already very harmful for the parasite, but then the double treatment, the parasite is completely dead. And it's very important because this concentration of DGAT inhibitor is not toxic, although this concentration, the host cells are completely fine. So that could be a way to think about affecting the parasite growth by these 2 treatments. Of course, the more important is to try to reach the chronic stage, the cyst stage of the parasite, for whom we have no drugs so far. We have identified that the cyst form that can be cultivated in vitro also form lipid droplets. You can see here, and they have cholesterol ester and triacylglycerol. And when we expose this cyst form with the drugs compared to the untreated samples, that the cysts are extremely small and they're terribly affected by this drug. And this is important because this drug, of course, is lipophilic. And at least we think that despite the fact that the cyst wall is very thick, at least it's allowed the penetration of lipophilic molecule that could be a basis for a new drug discovery. So as a consequences, by observing the parasite, not only inspecting its auxotrophy, we had some idea that we can either poison the parasite by giving some lipid analogs or other metabolite analogs, start the parasite by cutting the source, or overfeeding them because it will not stop ingesting some nutrients. The way to scavenge lipids is very unique. We believe vesicular, by using this network that is secreted and then make it fuse to the vacuolar membrane to trap host organelles, and non-vesicular route via the hijacking of membrane contact sites. The idea would be to identify drug targets in this uptake and storage pathway. At least we have identified some of them involved in the maintenance of the IVN or the processing of organelles like this lipase. And at least when you look at the storage pathways, is very vulnerable to DGAT and ACAT inhibitors. And that is very promising to fight against Toxoplasma infection in vivo and in vitro. So I want to thank many persons in my lab, including Julia Romano for this beautiful work on rat proteins, and all the other person who has followed the lipid transfer inside the vacuole. Of course, we have many collaborators who help us for this beautiful work. Many reagents proving from different mammalian cell biologists, and of course, the funding from NEID and American Heart Association. I thank you very much for your attention, and I'm glad that I am on time.
Thank you very much for a beautiful talk. So one question is, what are the roles for the IVN in the bradyzoids?
In the what?
In the bradyzoids.
That is a good question, because when the parasite converts from tachyzoid to bradyzoid, the IVN seems disappeared. But if you look at the cyst wall composition by electron microscopy, we could see that the cyst wall contains some kind of vesicles. And these vesicles, if they fused, they can form some kind of tubule, allowing the content of the host cytoplasm to be delivered inside the vacuole. But if that is the black box in the, let's say, nutrient uptake process of the cyst form. But obviously, because the parasite, the cyst form contains cholesterol. Of course, you cannot synthesize cholesterol. You must have a way to manage to retrieve cholesterol from the host cell. But looking by EM, there are no IVN in the, let's say, the matrix corresponding to the vacuolar space of the cyst wall.
So you said the parasite basically needs to scavenge these host lipids all the time. What happens during the chronic infections in the dormant phase?
That is
during the chronic infection and when they're in dormant phase.
So the question is that how they can retrieve nutrient, right? That is linked to my first answer. Nothing is known except that they must scavenge purine from the host cells because they cannot synthesize purine. So how the nutrient in general traversed the cyst wall, that is not known. It could be a very good project.
Great talk. In the IVN knockout parasites, do glycolysis or mitochondrial functions increase to compensate the nutrient deficiency?
That is not known. Yeah, Achille is not known this. No, this knockout is a beautiful tool, of course, to like a negative control for all our assays. Well, I can say that he has a normal grow in vitros. Of course, he's a bit stressed because he had this accumulation of amylopectin granules that are source of glucose. So it's possible that there's some kind of transient phase to the glycolysis because he has these lipids. But I'm not aware about study who are studying the mitochondrial functions versus glycolysis in the double knockout versus the wild type.
Any more questions?
I hope you like Parasite now because they're just amazing. And when I have a student, postdoc in my lab, I like to tell them you have to spend many times at the microscope, even the wild type, because you can discover by open your eyes so many unique organelles, so many unusual structures, and that could be very inspirational for new grant proposal. And I never start, for example, a project by a knockout or omics studies because maybe I think it's me, of course, that is not too intuitive, but microscopic observation is my way to go and to start a project.
Isabel, Akhil, great talk. Really enjoyed this very much. And beautiful, beautiful images. The question that I asked was about the IVN in bradyzoids.
Mm-hmm.
So if you have the IVN knockouts, do they work? Do they still form bradyzoids?
I think so. Yes, yes. There is one study who has from the Sible lab injected. I don't know. Okay, what is important is that we have different strain of toxoplasma, type 1 and type 2.
Right, right.
And only type 2 forms cysts in the brain. And I know that in the Sible lab, they had infected mouse with the knockout grade 2. I don't know if it's type 1 or type 2, but for I think it's type 2 because they look at the brain and they have said that there is maybe less cysts, but it was based on a very small amount of cysts that has been counted. So the answer is yes, I think, according to this unique study, that this double knockout can form cysts and transform into bradyzoid.
Yeah, because I was just wondering, one of the possibilities for not this knockout, but some of the other knockouts, especially the AGAT and the DGAT, ACAT and the DGAT knockouts, whether or not those would be good potential vaccine kind of parasites. Do they provide long-term protection if you just infect with those parasites in mice?
That is the next step. Yes, we have now the strain DGAT and LCAT knockout in the type 2 strain. And we have just the construct with a different tracker like luciferase to infect mouse and to see how the parasite can survive. But even in vitro, they're very, very weak. And what is interesting is that the high vulnerability of the parasite for this inhibitor, DGAT1, is because Toxoplasma has only one DGAT. And mammalian cells has two DGAT, 1 and 2, and is less sensitive to the inhibitors. So that is a very gold mine, this DGAT situation. But my goal is, of course, identify drug targets. After that, the drug design, it's another collaboration that I have.
A couple more questions. From Toby, is there any homology between the mechanisms Legionella used to recruit ER to the vesicle that used by Toxoplasma?
Another organism than Toxoplasma that recruit the ER?
Legionella.
Yes, of course, Legionella, indeed. Also chlamydia. No, that is something that is, I would like to emphasize, that is very interesting to be an ASM member, is that I like to let's say, compare the infectivity at the intracellular level between Toxoplasma and bacteria like Salmonella, Chlamydia. And we can see many beautiful examples of convergent evolution. By the way, they recruit organelles, also ER, multivesicular body, they cleave also the Golgi. So they act actually the same. And indeed, Chlamydia, it has been known that they recruit host membrane contact site, and we believe also is to retrieve lipids. So it's a kind of common themes between many intracellular microbes, parasite or bacteria. But of course, what is unique is the microbe effector that is on the way.
And from Hang Jun, does the liver stage Plasmodium falciparum mimic Toxoplasma in terms of the vacuolar extension and nutrient uptake?
That is a good question. And indeed, like Toxoplasma, Plasmodium liver stage form this long extension of the vacuolar membrane that also pervade all cytoplasm. But there are no connection between this extension for host organelle recruitment or lipid uptake, but it has been found out in the lab of Wulker in Germany that this extension is actually to, let's say, trigger the attack by autophagy from the protein ATG8. So somehow the parasite is projecting this extension to mediate an autophagy lysis outside his body, outside the vacuole. So sacrificing some portion of membrane to trigger and to attract ATG8, and in this case, the parasite will not be degraded by autophagy. So no, and you know, for example, for Salmonella, a lot of CIF extension also to interact with different host effector, but there are not that much study of all this extension derived from the vacuolar or inclusion membrane. But it's very fascinating, even in terms of biology and lipid metabolism. We really want to know the composition of this extension, but that is not possible to be purified.
Okay, no more questions. Thank you very much.
Thank you.
There's one more question. Dear said, is the vesiculation of the tubules in the PV essential, since it seems that the tubules could deliver lipids the same way?
Okay, you have to read again.
Is the vesiculation of the tubules in the PV essential, since it seems that the tubules could deliver the lipids the same way?
So the vesiculization of the what?
The tubules.
So the tubules are generated inside the parasite and secreted as a tubule. They will not be vesiculated. But at one point, yes, and understand when you have vesiculization, they will be detached from the vacuolar membranes to be dragged more inside the vacuole, yeah. So this process is kind of concert activity between the parasite and the host cells, because the parasite is recruiting the host cells machinery. But this tubule can be extremely long. So we believe that is because the parasite has to have this host organelle closer to his cell body in the middle of the vacuole when you have the concentration of the IVN, when you have enzymatic activity. Maybe it's not essential, Toxoplasma being famous to be redundant in many pathways. But what I would like to convey as a message is that the parasite is still debatable. He has some kind of endocytic activities. But I believe that the parasites secrete a lot of digestive enzyme inside the vacuole, proteinases, lipases, and also inhibitor of proteases to be protected. So meaning he has to process the nutrient inside the vacuole. So the vacuole can be considered like a massive external digestive compartment. And after that, the parasite, after that, from this host organelle, nutrient can be released and then reach transporter of the parasite at the plasma membrane and be internalized by active transporter. So I think this IVN is interesting biochemical platform. Many enzymatic reaction, and I mean, there are no other organism who is doing this. Maybe my lab has a lot of imagination, but something happening to this platform.
Great. Thank you very much.
Thank you. You can be reach me if you need me.
Yeah.